the world accepted those papers and the pictures that he did present as being evidence of a new virus?
Well, the world accepted these pictures, and the world accepted that Montagnier proved the existence of HIV, but this is what we have not accepted from the very beginning. We've been questioning, we've been asking in scientific papers, and today neither Montagnier nobody, or anybody else has responded to these questions.
Isn't there a chance that you're wrong, since everybody concedes that Montagnier did isolate HIV and purify it?
Well, who's seen it? We're giving you the evidence. And it's, to me, this evidence, and to my colleagues, this evidence does not prove the existence of HIV. Now, most of the people may have not even read the Montagnier paper, and yet they accept that Montagnier proved the existence of HIV. We mustn't forget that doctors are very busy, and it is very hard for everyone to go and study in detail all the claims. You have to really put a lot of effort to put all the things together.
But it's been 26 years since Montagnier published his papers. In 26 years, billions of dollars have been spent on HIV research. So doesn't the fact that, isn't Montagnier's paper kind of antiquated? I mean, hasn't it been proven that HIV does exist in the past 25, 26 years?
Well, nobody has presented any better evidence for the existence of HIV than Montagnier and Gallo. Gallo presented similar evidence. Apart, the difference between Gallo and Montagnier was that Gallo used, instead of umbilical lymphocyte T cells, he used the leukemic cell line. But otherwise, they published the same findings. And their findings today, still even today, are the best evidence anyone has presented for the existence of HIV.
So let's go to the test tube. What did Montagnier find in the purified band?
Montagnier took the proteins from that band, which he said, or the material he said was purified HIV, and then he reacted them with sera from his patient. And in the purified material, he found 3 proteins which reacted with antibodies which were present in the sera of his patient. P80, P45, 41 now, and P24. He said 41 was a cellular protein which contaminated his virus, made no mention of P80, and he said P24 was viral. Well, the question is, why only P24 viral and not P80? From antibody-antigen reaction, you cannot prove the origin either of the antibody or the protein. And he did not have evidence that P24 was coming from a particle. In fact, we did not know that there were any particles there. And then he said, he defined it by this reaction that P24 was viral and the antibodies were viral. This cannot be done scientifically.
HIV is said to have 9 proteins.
Yes.
But Montagnier only found 1 protein in his purified culture.
That's one of the big questions. If what he had there, what he called purified virus, it was HIV, and so it will have had all the proteins which a virus has, which HIV has, 9 or 10 proteins. And the patient, the antibodies which reacted with the P24 should have been also antibody to all these other proteins. And yet, the patient did not have any antibodies to react to the other proteins, and we don't know if he had any of the other proteins. So this is a big question. You cannot have a virus which has only 1 protein. The question is now, if that was a protein, if he had purified virus, where were the other proteins? And if the patient was infected with the virus, where are the other antibodies? How can we have only 1 protein and 1 antibody? it is not possible. But unlike Montagnier, who considered P24 the only HIV protein and 41 as being a cellular protein, Gallo in 1984 considered 41 as a more specific HIV protein and not a cellular protein.
So maybe Montagnier was just wrong and Gallo was right.
Unless you know that you have a purified virus, unless you have evidence that there was a purified virus, you don't know which one is right, because Gallo did not have any electron micrographs. Gallo could have said that 41 is HIV if and only if he had published electron micrographs and showed that what he called purified virus had nothing else but virus-like particles. Now, the next criticism is that Montagnier's particles, he said that his particles are typical type C viruses. Then, in 1984, he said that his particles were type D particles. In Levy, at the same time, said that they were type D particles. Gallo, again in 1984, said that the particles he's seen were belonging to the family to which his previous viruses existed. And so, and they are type C particles. The two retroviruses Gallo claimed to have discovered, they were type C particles. And then, from about 1985, 86, most of the HIV experts start saying that HIV is a lentivirus belonging to a totally different subfamily of retroviruses. And even today, there is still disagreement as to what subfamily and species the HIV particles belong.
Anywhere you travel in the world, you're going to find somebody that generally looks like somebody else. And when I look at electron micrographs, all viruses look the same to me. So is there really a difference between whether it's a C or a D or a lentivirus? Is it that big a deal?
Well, there is a big deal because they belong to different, according to retrovirologists, they belong to different, not only to different species, but to different subfamilies. And this is not different than saying that people saying that they see, one and the same thing, and yet what they see, some see a human being, others see a chimpanzee, and others see a gorilla. The difference is that much. We all belong to the same family. Chimpanzee, humans, and gorillas belong to the same family. All the retroviruses belong to the retroviral family. But we are in different species, and so are the HIV particles.
Would a trained eye be able to look under the microscope and easily distinguish between a type C, a type D, and a lentivirus?
That's what they say. That's what the electron microscopy say.
Using the electron microscope, how easy is it for you to differentiate between all these retroviruses? Because they all look, to someone like me, the untrained eye, they all look the same.
Certainly not. I will be able to teach you within half an hour. We have made a nice summary on these structural differences. You can measure. And if you start measuring the ribonucleoprotein, that's a complex of the genome and some protein, where is it in the virion, in the budding, in the immature particle? How is it organized in the mature particle versus the immature particle? What about the glycoproteinops? These are questions that can be quantitatively assessed. So you really can make an objective diagnosis.
So for someone like you, it's easy to tell the difference?
Yeah.
The cone-shaped core is very identifiable. It looks very different than a C-type, is that right?
Yeah, absolutely.
Okay, or at least to the trained eye.
No, you will see it.
Oh, okay. How come, in your opinion, did Gallo and Montagnier see C-type instead of lenti when they looked under their electron microscopes?
Because they had it in the brain.
Oh, they saw what they wanted to see?
Yeah.
Oh.